Ever wondered how a single cell could transform into a complex organism like us humans? This fascinating journey from simplicity to complexity is orchestrated by a process known as morphogenesis. Picture it as an expert sculptor, meticulously shaping an organism from a single cell into a full-fledged life form. In this grand design, two types of cells play leading roles: epithelia and mesenchyme cells. Imagine these cells as the bricks and mortar of our bodies. Epithelia cells, the bricks, are tightly packed together, forming the protective outer layers of organs and structures. On the other hand, the mesenchyme cells, the mortar, are more loosely arranged and often fill the spaces between organs, providing support and structure. These cell types interact in a beautiful dance, working together to form the complex structures of our organs. It's a marvel of nature's engineering, a testament to life's inherent drive towards complexity and diversity. Now that we've established the basics, let's delve deeper into how specific organs form. Imagine the brain, the command center of our body, but how does it form? Well, a significant part of the answer lies with neural crest cells. These are a group of cells that emerge from the most dorsal part of the neural tube during the formation of our nervous system. Picture them as the master builders, tirelessly working to construct the complex structure that is our brain. During the early stages of development, these neural crest cells are like undecided voters. They haven't committed to becoming a specific type of cell yet. But as development progresses, they start to migrate to different parts of the embryo. Now you might be thinking, migration? How do cells pack up and move? Well, it's not like they have little suitcases and passports. Rather, the process is driven by complex molecular and cellular mechanisms. These cells move along specific pathways, almost like they're following a map. Once they reach their destinations, these cells start to differentiate or specialize. It's as if each cell finally picks a major in college and sticks to it. Some become neurons, the building blocks of the brain. Others transform into glial cells, the supporting cast that helps neurons do their job. Some even become the cells that make up the adrenal glands, small organs that sit on top of our kidneys and produce hormones. But wait, there's more to this story. Neural crest cells are also instrumental in forming the accessory organs of the brain. These include structures like the pineal gland, which helps regulate our sleep patterns, and the pituitary gland, often referred to as the master gland, because it controls the activities of many other glands in the body. In essence, neural crest cells are like the ultimate multitaskers. They're responsible for the formation of many different types of cells and structures, each playing a crucial role in the functioning of our brain and overall nervous system. So that's how our brain, the most complex organ, takes shape. Our eyes, the windows to our soul, but how do they come into existence? Let's dive into this fascinating journey of ocular development. The formation of the eye is a complex process that begins early in embryonic development. It's like a symphony, where each instrument, or in this case, each cell type, has a specific role to play, and they must all work together in harmony to create the final product. Imagine a flat sheet of cells called the ectoderm. This is like a blank canvas, ready to be transformed. As our body starts to take shape, a portion of this ectoderm thickens and forms the eye field, much like an artist sketching the outline of a portrait. This eye field then splits into two, each budding off to form the optic vesicles, like two flowers sprouting from a stem. These vesicles push into the surrounding tissue, the mesenchyme, and start to fold inwards, creating a pocket. This is the optic cup, the basic structure of our eyes. Now, the magic of cell differentiation begins. The cells at the back of the optic cup mature into the retina, the light-sensing layer of the eye. It's like the film in a camera capturing the images we see. Meanwhile, the cells at the front of the optic cup form the iris and the lens, like the aperture and focus of a camera, controlling the amount and direction of light that enters the eye. But the eye isn't an island. It needs a network of blood vessels and nerves to function. So cells from the mesenchyme form the choroid, the vascular layer of the eye, and the optic nerve are connection to the brain. This intricate dance of cells, each following its genetic blueprint, creates the sophisticated organ that is our eye. It's a marvel of nature, a testament to the complexity and beauty of life. And that's how we get our eyes, the organs that let us see the world around us. So, the next time you look into someone's eyes, remember the intricate ballet of development that allowed them to see you too. The heart, our lifeline, but how does it develop? Let's delve into the fascinating journey of heart development. Picture this, a flat sheet of cells, seemingly unremarkable, but within it lies the potential to become the organ that will pump life through our bodies. This sheet of cells is part of the mesoderm, one of the 3 primary layers of cells in the early embryo. As the embryo develops, this sheet folds in on itself, forming a simple tube, much like when you roll up a piece of paper. This is the primitive heart tube, the humble beginnings of our heart. At this stage, it's nothing more than a straight hollow tube. But don't be fooled by its simplicity, for this tube is about to embark on an extraordinary transformation. Now imagine a piece of origami. It starts as a flat piece of paper, but with each fold it transforms into a complex, beautiful shape. This is exactly what happens to our heart tube. It starts to fold in on itself, not randomly, but in a highly organized and precise manner. This process, known as looping, is critical for the proper spatial arrangement of the heart chambers. The heart tube doesn't stop there. It continues to fold and divide until it forms the 4 chambers we are familiar with: 2 atria and 2 ventricles. These chambers allow our heart to pump blood efficiently, delivering oxygen and nutrients to every cell in our body. Even after the heart takes its recognizable shape, development is still not complete. The walls of the heart thicken, the valves form, and the heart starts to beat rhythmically and tirelessly, ready to support a lifetime of activity. Isn't it remarkable? From a simple tube to a complex 4-chambered organ, our heart is truly a marvel of nature. It's a testament to the power and precision of biological development, a process that occurs unseen within us, yet is so vital to our existence. And that's how our heart, the organ that keeps us alive, forms. Our alimentary canal, the system that fuels our body, but how does it take shape? Picture a bustling city. The buildings, the roads, the parks, all meticulously planned and constructed. Now imagine that city as our alimentary canal, a complex system built during our embryonic phase. Here's how it happens. During the early stages of embryonic development, a simple tube forms. This tube, much like the blueprint of a city, is the foundation of our alimentary canal. It goes from the mouth to the rear, and with time, different regions specialize to become the esophagus, the stomach, the intestines, and so forth. But a city isn't just buildings, right? There are power plants, water treatment facilities, and waste management systems. Similarly, our alimentary canal has auxiliary organs. These are the liver, pancreas, and gallbladder. They don't form directly from the tube, but from little outgrowths, like offshoots branching out from the main road. The liver, our body's chemical factory, starts as a tiny bud that expands and morphs into a complex organ. It's like a power plant, processing nutrients and detoxifying harmful substances. The pancreas, another offshoot, develops into a dual-purpose organ. It's like a water treatment facility, releasing digestive enzymes to break down food and insulin to regulate sugar levels in the blood. The gallbladder, a small organ tucked under the liver, is like a storage depot. It stores bile produced by the liver, releasing it when needed to help digest fats. These organs, much like the various facilities in a city, work in harmony to ensure the smooth functioning of our body, processing the food we eat, extracting nutrients, and expelling waste. Understanding the formation of the alimentary canal and its accessory organs isn't just fascinating, it's crucial. It helps us understand how our body works, how it develops, and how we can maintain its health. So that's how our alimentary canal, the system that fuels us, develops. So we've taken a whirlwind tour of how some of our major organs develop. We've delved into the intricate process of morphogenesis, unraveling the mysteries of how epithelia and mesenchyme work together to form our organs. We've journeyed into the depths of the brain, understanding the crucial role of neural crest cells and their accessory organs in its formation. We witnessed the miracle of the creation of the eye, appreciating the complexity behind the organ that lets us see the world around us. We ventured into the heart, the lifeline of our body, and its intricate development process. Finally, we explored the alimentary canal and its accessory organs, integral to our survival and well-being. Understanding these morphogenetic processes is crucial, not just for scientists and researchers, but for anyone intrigued by the marvel of human life. It helps us appreciate the intricacy and beauty of our own bodies. And that wraps up our journey into the fascinating world of morphogenesis. Until next time, keep wondering, keep learning,